students. Today I going to discuss about a very important topic, mitosis, which is from the reproduction lesson. First of all, what is mean by mitosis? So this mitosis is a process which will be occurs in all the living organisms for its growth and development. Not only for growth and development, so the repairing of the cell, the worn out cell, the damaged cells will be repaired by this mitosis process only. So this mitosis process, the name, the mitosis was coined by Walter Fleming, and the schematic mitotic division was explained by Theodore and Boveri. So now we'll enter into this mitosis process. The mitosis process, it is having majorly two stages. One is karyokinesis, another one is cytokinesis. Karyokinesis, the name, it has to be saying that karyos means a nucleus. Here, the division of nucleus, we are calling it as karyokinesis. The division of cytoplasm, we are calling it as cytokinesis. Okay, now coming to this karyokinesis, the nucleus, it has to going to divide and forms two daughter nuclei in this process. So in this process, it is having four major phases. They are prophase, metaphase, anaphase and telophase. Now we'll be going to discuss about one another. Okay, so first we'll be going to discuss about the prophase. What are the events occurring in this? We'll see one by one now. Okay, so first, the DNA, it has to be in the form of coiled, which we are calling it as chromatin. So this coiled chromatin, it has to condense. It has to going to fold it and it has to condense and forms a chromosomes, which can able to see under the light microscope. So first, the chromatin, it has to condensed into chromosomes. Then next, so we all know that in the interphase, so the DNA, it has to get doubled. So it means that two sets of DNA, it has to be present in the cell. Okay, so now what will happen? The homologous chromosomes means, so two sets. So it means that one pair, one pair it has to be present. So those homologous chromosomes, it has to going to paired. It has to going to paired and it has to attach at the centromere. Homologous chromosomes are going for pairing. Okay. So next, if you see that the nucleolus, inside the nucleolus is present. That nucleolus, it has to become smaller and it has to going to disappears. So nucleolus is going to disappears. Okay, then the nuclear membrane is going to disappear. The nuclear membrane is going to disappears. So then the centrosomes, it has to move towards the poles. It has to move towards the poles and it has to form the spindle fibers. So these are the events which are occurring in the prophase. So now the karyokinesis, it has to further move into the metaphase. So now we'll discuss about what are the events occurring in metaphase. In metaphase, so the chromosomes, it has to move towards the equatorial plate. So all the chromosomes, it has to arrange at the equatorial plate. The spindle fibers, it has to attach at the centromere. It has to attach at the centromere. And this centromere, it has to going to split and it has to separate the chromatids. So now all these chromosomes, it has to reach at the equatorial plate. So now next it has to moving to the anaphase.
Anaphase, the spindle fibers, it will be going for contraction, and the chromatids, it will move towards the poles. So the chromosomes, it has to get separated and it will be moving towards the poles. Next, we have to move on to the telophase. It is the last phase and it is exact opposite to the prophase. So why I am saying this in the sense, so whatever the events occurring in the prophase, it will be occurs as a reverse condition. So what is happens here? So the chromosomes, it has to decondensed and it has to become as a chromatin. Okay, the chromosomes, it has to decondensed and it has to turn as chromatin. And the nucleolus, it will be reappears. The nuclear membrane, it has to be reappear. The cell organelles, it has to be going to reappear. So all the events which will be occurs in the prophase, it has to be reversed in a telophase. Okay, after this telophase, the two daughter nuclei are formed. So now the cell, it has to enter into the cytokinesis. In cytokinesis, it has to differ in animal cell and plant cells. In animal cell, so the cell, it has to be at middle, it has to form a small pinch and it has to be going to elongate and it will be separate that cytoplasm and it has to form as two new daughter cells. Whereas in the case of in plant cells, so it will be having a separate process. So we all know that the Golgi complex, it has to form the cell plate. So this cell plate, it has to form at the middle and this cell plate, it has to divide the plant cell into two new daughter cells. Like this, the mitosis process, it has to going to completed. After completing of mitosis, two daughter cells are going to formed which are identical to the parent cell. Hope you people are understood the video. Please like and share the video and please subscribe. Thank you.